now. So nucleotide is mainly composed of three components: sugar, base, phosphate. We have discussed about the sugars in both DNA and RNA, that is deoxyribosugar in DNA, ribosugar in RNA, and nitrogen bases. We discussed about nitrogen bases, that is purines and pyrimidines. Purines are two types, adenine and guanine. Both adenine and guanine are present in both DNA and RNA. So purines are common in both DNA and RNA. Pyrimidines, so total pyrimidines are 3: cytosine, thymine, uracil. Cytosine is common in both DNA and RNA. So it is present in both DNA and RNA. So what are the bases which are common in both DNA and RNA? Adenine, guanine, purines, cytosine, pyrimidine. These three are common in both DNA and RNA. Now coming to the difference between the DNA and RNA regarding bases, that is thymine. Thymine is present in DNA, absent in RNA. Present in DNA, absent in RNA. Now coming to uracil. Uracil is present in RNA, present in RNA, and absent in DNA. So thymine is present in DNA, absent in RNA. Uracil is present in RNA, absent in DNA. Sugars, same. Ribosugar is present in RNA, absent in DNA. Deoxyribosugar is present in DNA, absent in RNA. So with this we have completed two components: nitrogen bases, sugars. Now coming to phosphate. Coming to the phosphate. So we discussed nitrogen bases and sugars. So, one type of sugar is present in DNA, one type of sugar is present in RNA. Some types of bases are present in DNA and some types of bases are present in RNA. But in case of phosphate, there is no difference in the phosphate. That is, phosphate is common for both DNA and RNA. So what is the structure of phosphate? The valency of phosphate is, phosphorus is 5. So 5 bonds, oxygen. So this is the structure of phosphate. So these are the three components present in the nucleotide. With that we completed the structure, completed the components of the nucleotide. So sugars, that is ribosugar and deoxyribosugar, nitrogen bases, that is purines and pyrimidines. So sugars and bases both are organic compounds. Phosphate is inorganic compound. That we should remember. So two organic components, one inorganic component. Two organic components and one inorganic component is present in nucleotide. Two organic components are nitrogen bases, sugars. Inorganic component is phosphate. So this is the structure of phosphate. Now coming to the structure of nucleotide. Structure of nucleotide. So we know that the nucleotide is mainly composed of three components: sugar, base and phosphate. Now we are going to discuss how these three components are interlinked in the nucleotide. So first we are going to discuss about the structure deoxyribonucleotide. So Is deoxyribonucleotide means definitely no oxygen at the second position. H. Here CH2OH. So this is the structure of deoxyribose sugar. Now if it is nucleotide, definitely there should be base and phosphate. So base, the nitrogen base, which may be either purine or pyrimidine, whatever it may be. So adenine or guanine or cytosine or thymine. No uracil, because it is deoxyribonucleotide. In DNA, no uracil. Thymine should be there, uracil should be absent. So A or G or C or T, any base, all four are bases only. So base. At the base is always linked at the first position of the sugar. This is the first position. Second, third, fourth, and this is the fifth position. Now sugar and base. So sugar and base is called nucleoside. What is nucleotide? If phosphate is added to the fifth carbon, then it is called nucleotide. So phosphate. Here CH2OH is there now. In hydrogen is replaced by phosphate here. So this is the base, this is the phosphate, this is the sugar. So this is the structure of deoxyribonucleotide. So this is the structure of deoxyribonucleotide. Here the valency is, oxygen is 2. So this is the structure of deoxyribonucleotide. Now coming to the structure of ribonucleotide. So what is the sugar present in the RNA? Ribose sugar. So first you can draw the structure of ribose sugar. So here OH, OH. So OH is present at the second position. Hydroxyl group is present at the second position because it is the ribose sugar. It is deoxyribose sugar. Hence OH is absent. So this is the normal structure of the ribose sugar. Now we are mentioning the structure of ribonucleotide. So sugar, base, phosphate. So base is always linked at the first carbon. So this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon. So here base should be there, that is adenine, guanine, cytosine, uracil. No thymine, because in RNA thymine is absent. Okay, this is the ribose sugar is present in RNA, absent in DNA. So uracil is here. Here in this case no uracil, because deoxyribose sugar is present in DNA. That's why thymine is there. No uracil. So base, sugar. Now it is nucleoside. So what is nucleotide? Sugar, base, phosphate. All three can be collectively called nucleotide. We are discussing about nucleotide. So phosphate should be present at the fifth position. So here OH, H is replaced by phosphate. P double bond O. So this is the phosphate. So now this is the structure of ribonucleotide. better you mention with this color, phosphate. So this is the sugar base phosphate, ribonucleotide. Now we are discussing the names of the bond between the sugar and base and sugar and phosphate. So very important. So sugar and base in the nucleotide always linked with N-glycosidic bond. So here the bond is there, no bond present. This is the sugar, this is the base. Sugar and base linked with N-glycosidic bond. Same here also, N-glycosidic bond. So sugar always linked to the base with N-glycosidic bond. Here sugar always linked with phosphate with ester bond. Very, very important. Here also ester bond. Phosphate always present at the or situated at the 5th carbon. Sugar always situated at the 1st carbon of the sugar. Here 5th carbon of the sugar, here 1st carbon of the sugar. Same in this case of ribonucleotide also. So phosphate is always linked to 5th carbon of the sugar. Here base is always linked to 1st carbon of the sugar. So sugar base. Sugar and base linked by N-glycosidic bond. Here also same, sugar and base linked with N-glycosidic bond. Sugar and phosphate, ester bond. Same here on, sugar and phosphate, ester bond. And sugar linked with base with the help of N-glycosidic bond at 1st position. And at the 5th position, phosphate linked with ester bond. We will learn names of nucleosides and nucleotides now. So first, what is nucleoside? Sugar plus base is equal to nucleoside. Nucleoside plus phosphate is equal to nucleotide. So sugar plus base is equal to nucleoside. Nucleoside plus phosphate is equal to nucleoside. Okay, so sugar plus base. In case of nucleoside, no phosphate. In case of nucleotide, sugar, base, phosphate. Now we will discuss different types of nucleosides and nucleotides. So nucleoside first. It's nothing but sugar plus base. So sugar plus, if the base is adenine, then it is called adenosine. Next, sugar plus guanine is nothing but nucleoside. So if guanosine is the base attached to the sugar, then it is called guanosine. If guanine is the base attached to the sugar, it is called guanosine. If the cytosine is attached to the sugar, it is called cytidine. If thiamine is there in the sugar, thymidine. Next, sugar plus uracil, uridine. So these are the different types of nucleosides. So how many nucleosides are there? Please remember, in case of nucleoside, no phosphate. So here no phosphate, Only sugar and base is called nucleoside. Sugar plus adenine, adenosine. Sugar plus guanine, guanosine. Sugar plus cytosine, cytidine. Sugar plus thiamine, thymidine. Sugar plus uracil, uridine. If you keenly observe all these different types of nucleosides, here adenine and guanine are purines. The suffix in purine is INE. So you can find INE here, INE here. Cytosine. So you should not mention the name itself of the pyrimidine cytosine is cytosine. So it is called cytidine. So and cytosine is purine or pyrimidine? Pyrimidine. Thiamine, pyrimidine. Uracil, pyrimidine. So what is the suffix? Dine, D-I-N-E. So here you can find D-I-N-E in this case. Okay. So nucleosides are of how many types? 5 types. They are adenosine, guanosine, cytidine, thymidine, uridine. These are the 5 different types of nucleosides. Now coming to nucleotides. So what is a nucleotide? Sugar plus base plus phosphate. It's called nucleotide. You know the difference between nucleotide and nucleotide first. Nucleoside is sugar plus base. Nucleotide is sugar plus base plus phosphate. So if the base is adenine, sugar plus adenine plus phosphate, then it is called adenylic acid. If phosphate is absent, only sugar and adenine is there, then it is called adenosine. If phosphate is there, phosphate is nothing but phosphoric acid. That's why you are adding the acid here. Adenylic acid. Next, sugar plus, if the base is guanine and phosphate, it is called guanylic acid. Next, sugar plus, what is the next base? Cytosine. Cytosine plus phosphate. Cytidylic acid. Sugar plus thiamine plus P, phosphate. Thymidyllic acid. Last base, sugar plus uracil plus phosphate, uridylic acid. So these are the different types of nucleotides. So there are 5 different types of nucleosides because 5 different bases are there, and there are 5 different types of nucleotides due to the presence of 5 different types of bases. So these are nucleotides, these are nucleosides. And one more thing you should remember, so adenine and guanine common, purines in case of both DNA and RNA. If sugar, adenine and phosphate is present in deoxyribose sugar, adenine and phosphate present in DNA, then it is called deoxyadenylic acid. If the sugar is deoxyribose sugar, then it is called deoxyguanylic acid. If the sugar is deoxyribose sugar, then it is called deoxycytidylic acid. In these 3 cases only it happens because adenine, guanine, cytosine, these 3 are common in both DNA and RNA. That's why deoxyadenylic acid, deoxyguanylic acid, deoxycytidylic acid are present in DNA. If it is RNA, adenylic acid, guanylic acid, cytidilic acid. So these are the different types of nucleosides and different types of nucleotides. Here also same, in case of nucleosides, 3 different types of nucleosides, that is deoxyadenosine if the sugar is deoxyribose sugar. If the sugar is deoxyribose sugar and guanine is there, then it is called deoxyguanosine. Same, if the sugar is deoxyribose sugar and which is attached to cytosine, then it is called deoxycytidine. So adenine, guanine, cytosine are common in both DNA and RNA. That's why when you are mentioning the nucleosides or nucleotides, you should mention deoxy if it is DNA and no need to mention deoxy if it is RNA. So, but in case of thiamine and uracil, no need to mention whether it is deoxy or normal. Why? Because thiamine is always present in DNA, uracil is always present in RNA. So if thiamine is there, definitely it is DNA. If uracil is there, definitely it is RNA. So there is no confusion whether it is in DNA or RNA. So in this lecture, we studied different components of nucleotides, structure of nucleotides, that is different nucleosides and different components of the nucleotides. Everything we completed. With this, I conclude the session. Thank you.